and a dermatologist as well, with research interests related to his presentation today. I think psoriasis, oncology, chemotherapy, side effects, among some others.
All right. Well, thank you very much. I just want to a little bit about my background. I mainly practice clinical dermatology with certain interests, as are outlined here. I do want to mention, I'll kind of go through how this talk even came about, but I am not an expert in anthropology other than what I've read about. And, this talk I've given to different groups, from undergraduates to internal medicine, grand rounds, and dermatologists as well. And I haven't given it to PhD students. There is definitely some genetics in here, probably even beyond what I understand. And you could probably teach me some things. But I think what the most interesting thing about this, I'll kind of, as I get through, we'll go through the story of how this came about, but is it involves all of us. I don't care what level of anything. For a human being, I find this very interesting. And what I'm going to really kind of map out today is, it's pretty well known that as primates, Homo sapiens are really the dominant hominid clearly on the planet. And of course, there's a lot of things you're going to read. We're dominant because of our ability to communicate, our ability to gossip. If anyone's ever read the book Sapiens, it's a really interesting book on anthropology. And I'm here to make a little bit of an argument. It's already been made. I've just kind of summarized some of the data, that the skin plays a huge role in how humans eventually migrated around the earth. And it was really changes in our skin that allowed for this to happen. Of course, there's many other things that had to be happening simultaneously. And as we go through the lecture, I'm not talking about a linear path. Clearly, there's things happening with enlargement of the brain, primates becoming bipedal that we'll talk about a little bit, but all these things are happening simultaneously. And hold on a second here. Sorry, guys. All right. So, one day I was in clinic and I was doing a biopsy, and I think it was a darker Hispanic young girl, and I took a shave biopsy of the skin. And I don't know why, I've been doing this for a while, but I took a little shave of the skin. And in this moment, I thought, geez, everyone's dermis looks exactly the same. And what we really, when we start to relate with each other, and it's in the news all the time nowadays about race relations and the politics around race and inclusivity and diversity, but a lot of it is encoded by this little cell, or the melanocyte, that's in the epidermis, right? I mean, depending on where we are in the skin, we're talking 20 to 200 microns of epidermis somewhere. And so from that, I kind of started looking into really the genetics and the evolution of the MC1R gene itself. And that kind of led me into other paths. And that's kind of where we are today. Okay. I'm going to mainly talk about some structures of the skin that have evolved over time, a timeline, how we know this a little bit, and then just some hypotheses for why things occurred. Clearly, no one knows definitively why some of these changes did occur. So quickly, it doesn't matter what living organism you are, especially if you're a mammal, we have these skin functions. I'm not going to highlight these, but clearly they're pretty self-explanatory. So I always like this picture right here. So this is Australopithecus, one of the earliest ancestors of Homo sapiens. And I'm going to also draw a linear line. We're not going to talk about Neanderthals. We're pretty much going to assume we're going to go from primates to early bipedal hominids into Australopithecus into Homo sapiens. So that's the linear line we're going to take. However, realize, of course, there's multiple branches that are involved in that. So here we have Australopithecus, a bipedal primate. And here, this is myself, and this is my daughter here in Arizona. And clearly there are changes that had to take place for Australopithecus of Southern and Eastern Africa to eventually evolve into the species Homo sapiens. And here, of course, we're a little bit more hairless. We are bipedal. And though I did not walk from India to Arizona, there was probably some migration that did have to take place. So when you really go back and we think about our primate ancestors, the big difference between, of course, primates and humans is the fact that, we're bipedal, and that had to take place initially. And then there's the question, well, why did that even happen? What's the advantages of being bipedal? Because when you really think about our quadrupeds, they are much faster. And in the game of you're going to have to chase prey and you're going to have to run away from predators, being fast is fairly important. So this is just a graphic here of Southern and Eastern Africa, and we're going to assume that this is where our basic roots are for all of Homo sapiens. And just a picture of the open savanna here. So advantages to bipedalism, right? So I would say that definitely as we became more bipedal, you definitely have greater visibility. We're not the only bipedal mammals. Kangaroos, talk about birds, we have ostriches. There are other bipedal organisms, right? For that to take place, of course, our hips had to shift downwards. The occipital foramen had to shift a little bit. We had to be able to rotate our head without our shoulders, multiple things. But one advantage of being bipedal is, of course, as our brain was enlarging, now we had the ability with opposing hands to use tools. We also had greater visibility. But really, the big question is why did bipedalism, if it slowed us down, why was there a pressure for bipedalism to occur? Because why weren't we being eaten? And though no one knows for sure, there is something called the endurance running hypothesis, in that bipedal primates, the way we would essentially hunt and increase our range was by wearing our prey down. So when we look at all mammals, humans actually have some of the greatest endurance running lengths of actually any mammal, unless you're swimming. Or potentially flying, but we'll just say swimming, like a whale. But so the thought was that we could wear so you're going to start in the forest. We're going to, during the day, try to leave, and we're going to hunt by running, wearing our prey down, and of course, bringing it back to the forest. Because really the challenges at the time were, not only were we bipedal, we're trying to expand our range, get more food to feed our large brain, but we're also limited because the savanna's hot. And as we left the forest, there's only so much time we had to hunt. So when we talk about Australopithecus, one of the first bipedal hominids that we know of, like Lucy, we may have heard that before, essentially lived in higher altitudes. There's definitely cooler temperatures at night, so they can go back during the day. These were forested habitats. They had fur very similar to chimpanzees, which did provide for warmth at nighttime. The challenges were that some of these things, especially hair, limited the hunting range, right? And one of the thoughts is either humans wanted to, or early hominids wanted to either expand their hunting range, or just because of climate change at the time, maybe a shrinking forest, where they were forced to leave as the forest shrunk, they were forced to leave the shaded areas to hunt during the day. So we did have inefficient cooling mechanisms, which definitely limited its range in the open savanna. So here we have our limitations: inefficient cooling in the open savanna. We did have thick fur at the time. We definitely had a lack of eccrine glands at the time, comparably to now, but actually very comparable to chimpanzees as far as the density of the Australopithecus eccrine glands. And brain tissue, our enlarging brain, is very sensitive to heat. So we didn't want to expand our range too far too quickly, because that could eventually result in heat stroke. So eccrine glands themselves had to change a little bit. Some of the genetics behind that we'll get to, but eccrine glands are pretty specific to mammals. And when we look at our old world apes, humans will include in that, gorillas, macaques, what we find is that humans actually have 10 times the density of their eccrine glands, which is kind of interesting. So by far, we are the sweatiest primate. So as a joke, whenever I see patients with hyperhidrosis, and the young kid with hyperhidrosis, just so everyone knows here, no one likes hyperhidrosis, but I try to express the fact that they're probably just evolutionarily advanced because they have just so many active sweat glands. But sweat glands themselves, the function, especially we have such a high concentration on the hands, is really for gripping and be able to hold on to things. So here's just one graphic. This is data from, I did not do this research, but this is just 3 slides of eccrine glands within 3 different old world apes. And bottom line is humans have the highest concentration. When we look at genetic changes here, so the engrailed 1 gene encodes for the density of eccrine glands in the skin. And the thought is essentially about 8 to 11 million years ago. So this is still when we're still in a forested habitat. The enhancer protein, so we have human enhancer here, the human ECE18, and this is chimp. They started diverging around that time. And the human the ECE18, the enhancing gene, is actually much more potent. It stimulates ingreld-1 to a greater degree, and we definitely have a greater density of eccrine glands in the skin. So we now have some sweat, so we can sweat more, but the problem is we need to evaporate that sweat, right? So as we're expanding our range outside of the forest, ideally we do not have hair to allow for evaporation of that sweat to take place. Well, essentially, if we look at almost all other mammals, most mammals are fairly hairy because there is a selective pressure for most mammals to retain hair. Now, whether that's for thermal regulation, I think there is a component of identification, mating, sensory, as well as camouflage, depending if you're predator or prey. Great picture of One Direction here. And clearly, I have young daughters, but I'm sure that's an old picture, but the hairstyles would be very appealing as opposed to if they were different. So mammalian hair does have a function. So why is it that we lost it? And one of those thoughts, of course, is that to improve our range and our ability to migrate outside the forest and to allow maximum function of our eccrine or our sweat itself, we wanted to increase evaporation. So where did it all go is the big question. Of course, nowadays we use laser hair removal, pay money to make that go away. There's a couple ways we know about it. It's pretty interesting because, so one is looking at lice data, and whoever thought lice were going to be interesting, but this is actually really fascinating. And then we're going to look at some genetics of the HR gene, and what we'll find is actually, how they date evolutionary changes in the HR gene actually correlate with some of the things we know about hair loss in lice. And I don't know if anyone here studies lice. Does anyone here study lice? No one knows, because it's actually really fascinating. So when we look back at lice and how do we know how lice help determine kind of when hair was eventually lost is, we're going to make the assumption, of course, we're closer related to chimpanzees than we are to gorillas. And chimpanzees and humans share head louse, right? So pediculosis. And gorillas primary, so even nowadays, if you're going to go into a part of Africa that had gorillas and they had lice, that's going to be thyris. So what we call the crab louse or the genital louse. And if you have a fully haired individual, as i.e. a chimpanzee or gorilla, and there's competition for resources, think of the hair like a forest. We have a huge forest. We have two populations. One's going to out-compete the other. So pretty much the gorilla itself strictly has thyris or crab louse, and the chimpanzee strictly has pediculosis. And we're the only ones who pretty much share both. And so if we go back, I'm just going to go to the next slide here because it's just interesting. Hopefully everyone can see the arrow here. So this is our crab louse, and this is going to be pediculous right here. And these are our early primate hominids. We're going to start here. We're going to see gorillas are primarily going to have thyris. We have our more chimpanzee. Early hominids are mainly going to have pediculosis. And the bottom line is somewhere at this point, an Australopithecus organism, this is just hypothesized, probably came in contact with a dead gorilla. And that's what this butcher marks is here. And there was a host switch. But that host switch could only take place if there was essentially an island of empty skin in between. So the hypothesis is at this point where there's a host switch, in between here is where hair loss is taking place, somewhere between 3 to 7 million years ago. And this is why humans have both, because there's almost like two islands of hair, and still chimpanzees who are fully hairy have pediculosis, and the gorilla still carries thyris. So who would have ever thought they were historically so interesting? And so when we look at genes and we look at evolutionary pressures on genes themselves, one gene that's implicated is the HR gene. So if someone has mutations in the HR gene is important in telogen to anagen switching. And so mutations cause alopecia universalis congenita. These are babies postnatal that are born with hair. However, after the first hair cycle, that hair does not regenerate. And HR mutants do cause a defect in Wnt signaling, which is important in hair growth. And so if we go back and we look at pressures on the HR gene itself, I'll go back. I'm going to go to this slide. I'm going to go back as well. So this is looking at the HR gene itself. And in this particular study done here, they're looking at rate of change within the gene. And what they found is at this point, the rate of change in the human HR gene was greater than the rate of change in the chimpanzee gene. At this point, it's assuming an almost identical gene. And therefore, at this point, there was a divergence that put a positive selection on decreased, or this particular mutation that caused decreased hair growth in humans. That was occurring approximately 6 million years ago. So here, essentially, I just have this written out where we have this analysis looking at where there's an increased rate of non-silent mutations probably occurring in that period about 6 million years ago, which completely separate is also happening right around that time period, around 3 to maybe 7 million years ago. So in that time period, we are becoming more sweaty, we're losing hair, and because of that, we're able to increase our range because we're able to cool our bodies more efficiently, and we're able to leave the forest and hunt, and then provide more food for our brain, which is once again continuing to improve. Any questions so far? Everyone, is this interesting at all to anybody? Perfect. So here's our challenge in the end, right? Now I'll go to this next slide here, but if we look at a chimpanzee, the skin under the chimpanzee, the true epidermis, is fairly fair. We can see the face. There is some pigmentation forming here. But if we lost the hair, well, this is going to be more of our natural skin color. It's going to be quite fair. And so at some point, the lack of fur, of course, we have more exposure to the elements. The one thing I do want to point out, if we go back, is just talking about exposure to the elements, is back when we're looking at the lice data, the body louse, so essentially the clothing louse, which is quite specific to Homo sapiens, does come from pediculosis. So at that point, the divergence from the head lice to body lice occurs about 40 to 70,000 years ago, which also correlates to the beginning of clothing use in early humans. Let me cycle back here. So lack of fur, we have the exposure to the elements, of course, sunburns, inability to tolerate low temperatures. At the same time, we have this increased sweating. Well, at some point, the lack of hair does become an issue, mainly because hair itself does provide for a ultraviolet protection factor, and the density and the thickness of the hair does increase hair UPF. So we're definitely losing UPF. So now we have this pressure to synthesize melanin, and we have this pressure on the melanocortin receptor 1 gene to start making more eumelanin, which also starts to occur somewhere between 6 to 7 million years ago. So I think the standard kind of question of, well, why did humans make eumelanin? No one really knows for sure. I think the gut reaction is, well, it's probably to prevent skin cancer. But if we look at the normal lifespan of early hominids to be 20 to 30 years, the chance of dying from skin cancer was going to be fairly low. So is there another reason? One of the current theories is looking at folic acid. And, folic acid itself does have a seasonal variation. And, there are a few studies that look at folic acid levels in the summertime are typically decreased compared to wintertime. And the bottom line is folic acid does absorb ultraviolet radiation, mainly in the UVB range, but also in the UVA range. And in the UVA range, because ultraviolet light does penetrate fairly deep into the skin's vasculature, it could theoretically be one of the reasons why there was a pressure to protect ourselves from ultraviolet light. Because if there is, of course, a decrease in folic acid, not only will neural tube defects be more prominent, but also folic acid plays a role in sperm mobility. And both men and women could have issues with fertility at that point. So one of the prominent theories for why we developed eumelanin is to protect ourselves from folic acid degradation. And you always have to wonder other theories, but we'll stick with folic acid. Or I keep theorizing. We already went through this. So there's all kinds of melanin. When we think about melanin itself, we also have eumelanin and pheomelanin as being the major kinds that are determined from the melanocyte itself. And And really that's kind of determined the type of melanin, whether it's eumelanin or pheomelanin, by how much cysteine is present within the cell and the activity of tyrosinase. And by having these mixes of mainly tyrosinase and activation of the MC1R gene and certain other little tubules, we can come up with all kinds of Fitzpatrick skin types. And it's kind of interesting. You have some people with blue eyes and darker features. We have blonde hair and blue eyes, and we have brown skin and dark hair. And then even in India, we're going to have even brown skin with green eyes. So all of these interesting mutations that kind of take place. And as I go through this, we'll kind of map out where these mutations are prominent, and some of the mutations responsible for some of the obvious things, more like blue eyes or blonde hair. So I think we've all kind of seen this here. I just like this basic diagram of the melanocyte here. So this is the agouti signaling protein here. This is an antagonist to the MC1R gene itself, the protein. And then we have alpha MSH signaling here, stimulating melanogenesis itself. Some things I do want to touch on. We are going to talk about selective pressures on the MC1R gene, but also just to point out these. So in addition, of course, mutations, of course, in MC1R are definitely responsible for the redheaded phenotypes. But we also want to talk about other genes that change the pH or the ion concentration of the melanocyte itself, often resulting in different changes in the activity of tyrosinase. So mainly focusing on these three genes that are commonly mutated in Northern Europeans to give different phenotypes. And then the KIT ligand itself. KIT ligand activation also increases tyrosinase activity, but mutations in KIT ligand can also be seen in patients with blonde hair. And of course, the purpose of eumelanin is to protect, well, we don't know for sure, but once again, theoretically, to potentially protect ourselves from folate degradation. So we kind of went through this. Don't need to go through this too much more, but we do see the sun-resistant alleles, so that's alleles, so the production of eumelanin do correlate with the loss of hair, and that's about 1.2 to 6 million years ago. And I think we go back to mutations in the HR gene, and that was happening roughly about 6 million years ago, at least the HR gene, and the one study looking at the evolutionary rate of that gene itself. So the interesting thing about this gene itself is that it's highly preserved in areas of Southern Asia and Africa. So there is definitely a positive pressure to maintain that gene. What does that mean? There are no other alleles that are really naturally present within Africa or South Asia, as opposed to all of the alleles that are going to occur in other parts of Asia and Northern Europe. So clearly, having pigment in a sunny environment is extremely important for survival. And whether, once again, that's folate or skin cancer, it's unclear. But once again, you do not have amino acid variants in Africa, India, New Guinea, which implying a strong functional constraint on the MC1R gene itself. Going back here, we're going to touch on this later, but loss of function. So once again, we're going to talk about patients with blonde hair, fair skin is tolerated in northern latitudes. And we'll touch into this as well, but there is no current evidence that the diversity that is seen in the multiple alleles outside of Africa has been enhanced by selection due to the vitamin D hypothesis, where essentially, I think we're all aware of that northern latitudes, we have to lose pigment, so we have increased production of vitamin D, but there is no evidence that the diversity is related to that. And we can touch a little bit, we don't know that for sure, of course, but we can touch on maybe what could be responsible for increased vitamin D production or more efficient vitamin D production in northern latitudes in a bit. So we're going to first kind of just focus on the MC1R gene itself. We already know that mutations in the gene itself are associated with the increase in pheomelanin. I'll go to the next gene here. This is just talking about very specific in Irish individuals or Swedish individuals. Mutations within these very specific point mutations here are commonly found. But this is actually even more interesting. So this is about migration out of Africa. So partially, and this kind of goes with the maps, we kind of assume that humans kind of started migrating out of Africa about 50 to 100,000 years ago. But we start to see variations or new alleles showing up about 100,000 to 250,000 years ago. for MC1R variants encoding for pheomelanin, causing pheomelanin, of course, 80,000 years ago. And then once again, why there's a lack of pressure that allows for the diversity in the alleles is unclear. Maybe we just didn't need the pigment, and it was therefore over time people in northern latitudes lost it. So this is just going back, and this is just looking at years ago. This is mainly going to be more Australopithecus, early hominids leaving Africa here. And so this is kind of in this period of time is where we start to see these MC1R alleles starting to show up. So I did want to touch on a few of the other encoding proteins. One of them, I think in that original slide, just linked to melanocyte. This gene right here encodes just an ion transporter, and this is another transporter protein, but mutations in both of these genes are almost exclusively seen in individuals from Europe. So once again, that we talk about mutations in the MC1R gene itself is causing different phenotypes and skin color. Beyond that gene itself, there's other mutations that are occurring that cause changes in tyrosine, why am I blanking? Tyrosinase activity, sorry, also resulting in different skin colors here. So this particular allele right here of this protein is fixed in almost all Europeans and absent in populations without European ancestry. And this particular allele here is also associated with European ancestry. KIT ligand itself also stimulates tyrosinase. This particular allele here is what leads to blonde hair. And so we can definitely have mutations in one place and not the other, and then we start to get these different phenotypes. We haven't talked about OCA2 mutations resulting in blue eyes as well. So it's interesting when we look at just changes in the hunter-gatherer population versus farmers. Traditionally, one of the earliest thoughts, because if we look at the Alaskan natives, they still retain their pigment. And one of the thoughts is that it's because they receive most of their vitamin D from their food source or salmon. Therefore, there was really no reason for them to lose pigment, which goes against what I said earlier, as far as that maybe pigment has no role at all in vitamin D metabolism. But it is interesting that our Western hunter-gatherers often had more dark features compared to our agricultural farming population. Going back, I just want to point on some of these mutations here. If we go back to this bottom end of the slide here, we got the origin of blue eyes, which mainly occurs with the OCA2 mutation about 42,000 years ago. Then we talk about light skin. Those genes start to appear mutate about 22,000 to 28,000 years ago. And then once again, we see these different permutations of these genes in Northern Europeans, mutations here, resulting in light to intermediate skin tones, as well as blue eyes. And then we can even go over here where we have mutations in KIT ligand resulting in blonde hair, as well as light skin here. Then we have dark skin, blue eyes, of course, patients or people with OCA2 mutations. So you can kind of see how these different mutations took hold in different parts of the world, resulting in different phenotypes that we all see nowadays. Once again, this is also looking at gene mutations. So we can see this is just a evolutionary model. We see mutations in KIT ligand that are nearly absent in Africa, but present in a lot of Asia, as well as Europe, and really once again, absent in South Asia here. Mutations in OCA2 resulting in blue eyes. We can mainly see that in Northern Europe, and also these functional MC1R mutations right here. We can see those as well as in Asia, which is kind of interesting. So it's kind of neat. You kind of know this information now. And so this is my daughter here. Clearly has a pretty solid MC1R gene making eumelanin. We could see her friend here with likely who has a OCA2 genotype, and we have darker hair with fair skin, so Northern Europe, and then this is our soccer team here. And it's kind of interesting to start to pick out where some of these mutations are. So as I was touching on before, there really is no strong evidence that the loss of mutations within the MC1R gene, where there was a pressure because of low vitamin D. Clearly, having low vitamin D would result in rickets, and those individuals do not survive to reproduce. But it's pretty interesting here, as we really look at skin anatomy, majority of vitamin D production is occurring in the stratum spinosum, right in here. And about 70 to 80% of our pigment is mainly in the basal layer of our skin. So yeah, there is maybe 20 to 30% of pigment that's in the stratum spinosum, but as we're getting irradiated with ultraviolet B to make effective vitamin D, our melanin is not necessarily shielding us. And so, the relationships between, though, yes, pigment may reduce our levels of vitamin D a small amount, there's no strong evidence that lack of pigment is the causative reason why we lost pigment as we started migrating into Europe itself. This is just a nice slide, just looking at some of these mutations, keratinocytes producing KIT ligands, stimulating melanogenesis, and these are some of the mutations that we're going to see in Northern Europeans all through here. So the one thing I do want to point on, and this is, we're going to look at this particular gene right here, DHCR7. This is an important gene in vitamin D metabolism. Mutations within this gene do cause an increased substrate within the epidermis and actually result in more efficient vitamin D metabolism. So we do have these genetic variants that actually are found in Northern Europeans. And the bottom line is patients with these genetic mutations more efficiently make vitamin D. So one thought is that patients in Northern Europe, yes, there is less ultraviolet light, though they have less pigment, it was more the fact they were able to make vitamin D more efficiently because they had enzymes that were more efficient at making that vitamin D within the skin. If we do look at this particular map, and we're going to look at mutations in this DHCR7, this is this yellow area here, we can take a look at this particular variant in the British population, the Finnish population here. We can look at it in Northern Africa here, and you can see that in more northern latitudes, there's definitely these variants that result in more efficient vitamin D metabolism. And just interestingly enough, particular mutations within this gene also put us at risk between type 1 or for type 1 diabetes and MS. And the interesting thing is, as you really go back and you look at vitamin D itself in invertebrates, we tend to think of vitamin D and we think of the bones, of course. And I think we're learning more and more of its large role in immunity itself. But in invertebrates, the vitamin D itself plays a large role in maintenance of the immune system itself. So. All right, here. So where are we at? About 35. So in general, I'll kind of come to the end right here. I did shorten a lot of slides out of it, but the argument I wanted to make is, as we think about the melanocyte itself and the pigment we have in everyone in the room today and around us, it's really diversification of the melanocyte due to migration and environmental pressures. And the migration itself resulted in changes in the skin, and the changes in the skin have allowed for humans to successfully migrate around the world and allow our brain to adapt and become better at speaking and recording history. And in the end, we are one of the most successful species on the planet Earth. And hopefully that'll continue. I worry about that a little bit. So in summary here, the skin played a crucial role in the human migration out of Africa with key changes in pigment, hair, and sweating. Vitamin D status may evolve independently of skin color by adaptations in vitamin D metabolism. MC1R variants evolved likely because of a lack of selective pressures in northern climates. We just talked about the MC1R gene. I'm not going to recap that. And several genes are involved in human pigmentation patterns. They can be traced back to human migration around the planet. That's it, guys. That's my lecture for today. Any questions?
Thank you. We applauded that we were muted for most of it. So.
I hope it was interesting. Did I go over information that people already knew?
No. I see shakes. I think this was a lot of new stuff. Yes.
I have two questions.
Yeah. Help yourself. Probably, Neil, I guess you could. You maybe can't hear from the back of the room.
Can you hear from here?
No, I can. Yeah. I'm just getting close to my computer.
Oh,
okay, I can come closer.
yeah, come on up.
Thank you, Dr. Neel, for the well-prepared presentation. It was very interesting. And I actually had two questions. So the first question, I'm going to ask you to ignore the ethical part of it, but it's just something that I'm coming from Kurdistan, Iraq, and I've heard that there are parents who wants their children to have certain features, their newborn children to have certain features. For example, have you ever, I don't know about it, maybe you do, if there were anyone who tried to have mutating the genes, for example, in my country, blonde and blue eyes are very desirable. So if someone comes and say, oh, please, can you mutate the MC1R gene for me so my newborn can have lighter skin or something like that? This is for my first question. And and actually, have you had any trials with any source of animal to have certain features in them? I know this might not be ethical, but it interests me that people can do that. And the second one is, did the migration had any effect, or do you think that this migration that now we have, for example, I live in America right now, do you think it will have any problem with tracing the genes to its origins? These are my two questions.
Yeah, I think those are great questions. So the first one is interesting. I don't know of any current like CRISPR studies, anything where someone is able to, and I'm sure it's happening because I think it makes perfect sense. And I'm sure there's someone potentially working on it, but I don't know anyone who is doing that. It is interesting that you say, I was thinking back for, what are really, there was no selective, in other words, the MC1R gene is so well conserved in Africa, right? And it may be this folate hypothesis. One thing I was also thinking was, though, people with like oculocutaneous albinism in Africa are shunned, right? And, maybe, as humans in that time period we're very gossipy things, and we communicate and we form opinions, and really no other animals do that we know of. Of course, we can't communicate that well with other animals, but I always have to wonder whether people are just shunned who had these mutations, and whether that was part of the pressure on that gene to be so well conserved is kind of human society and what we want. And I know this is not what you asked, but kind of even in India, we kind of see the idea that to be fair skinned is to be of higher status, right?
Exactly.
Your second question, I think you're right. I think, now there really isn't a selective environmental pressure on pigment. It's more, I think, the social factors of how we blend genes, what do we find attractive, and who are we going to mate with? And we're going to end up with all of these really neat permutations of all of these basic genes, which may cloud the picture a little bit. But you're right. I think, of course, as time goes on, it's going to be harder to maybe find the origins of where people came from.
I guess I would add maybe, as far as like choosing what your offspring is going to look like, maybe in the, I mean, just hypothetically, and ignoring the ethical issues, there is for infertility, for in vitro fertilization, there is the opportunity to test for, genetic mutations, parents who have, really carry deleterious gene mutations. You could sequence in for those and then not use those developing embryos for the in vitro fertilization. So in theory, you could do that for skin and hair color too, right? I mean, I guess, but I mean, that's been the subject of some movies. I can't quite remember like which ones they are, but I know there's definitely been science fiction movies that talk about that idea, which is scientifically, medically would be
Feasible.
possible, but you might end up with somebody who has something
If they go.
much worse than having the wrong, quote unquote, wrong colored skin or hair or eye. And for the animal models, I mean, definitely there are mouse models with all of these mutations. In fact, a lot of the work done for the agouti and the hairless HR gene mutations comes from mouse work. So, I mean, there are agouti mutant mice that are a beautiful golden color, not to discriminate against the other strains of mice. Or, you know, a lot of albino strains of mice too, right? That we use in research. And hairless mice are common in skin cancer research, the HR mice too.
Great.
Yeah.
Thank you so much, Dr. Neel.
Yeah,
Thank you.
thank you.
Okay. Anybody else? I don't know. Is this an area where you see a lot of current research or papers?
I
Still,
don't. I really don't. You know, a lot of this is done by, I think, our anthropology group. There's not very much, almost none, in clinical dermatology, I'll tell you that. And I just find it so current. I find, you know, it's what we deal with every day. And especially with skin cancer, knowing some of these mutations that make ourselves prone to it. And I just find it fascinating.
Yeah, I would have said before you started talking that if some student had asked, that the vitamin D hypothesis was probably the reason for, you know, decreasing pigmentation in Northern European populations. But as soon as you said it, that makes perfect sense, right? You know, you're not going to have skin cancer by age 20. I suppose you could have horrible sunburns. That might be something, but that makes sense. You guys are quiet. We had a room full of people interested in skin and skin cancer research.
Cool.
I have a question, Dr. Patel.
Yeah.
I was interested in sort of the part of your talk about.
Maybe introduce yourself, I think.
Yeah, I'm Justin Rudd. I'm an MD PhD student working with Dr. Hansen on skin biology. So very interesting talk. I had never heard about most of this anthropology side of things, but I was interested about the portion of your talk about hair follicle loss, and you mentioned those mutations in the HR enhancers that sort of, you know, change telogen to anagen transition time. I'm wondering if there's anything known about kind of the loss of actual follicle number during that transition, or whether everything we know is mostly related to, hair follicle transit times.
Yeah, I think it's more the trans. So if you really look at the density of gorilla hair, it's about 60 follicles per square centimeter, and that still transitions even to humans. They're probably just more vellus hairs. So I think it's more miniaturization of those follicles. The densities are roughly the same between early primates, or at least not early, just primates.
Okay, interesting.
Can I ask like more on the anthropology side of the things? So you mentioned that like there were like surface pressures. Can you hear me?
Introduce yourself.
Oh, sorry. I'm Fred. I'm a PhD student working with David E. I'm more on the hearing side, but I guess the question is related to like you mentioned that the pressures for different variations in skin across where you live. Do you think that same pressure exists in the modern age, or do you think that like the confounding kind of backgrounds of people in these types of pressures will result in new phenotypes, or like how do you expect those pressures to change in the modern age?
Yeah. So I think the pressures we're going to talk about are going to be both pigment as well as vitamin D pressures, right? Now, the thing is so in sunny environments, this is what I was looking into. So if the folate hypothesis, where are we going to find issues, right? So we have this folate hypothesis, and if you're fair skinned and you live in a sunny place and you're female, it's definitely something to think about. In other words, yes, we know in the summertime there's less folic acid, or there's less serum folic acid. And there are a few case reports of decreased folic acid or serum folic acid levels in patients who use tanning beds, right? So I do think, of course, if that's an issue, then yeah, we can supplement with folate. But I think, now we have fair-skinned individuals in really sunny climates because we're moving, not by walking. And then, as far as pressures, I don't see the pressure to make pigment coming back anytime soon in someone fair-skinned, maybe as we all blend together into one race, maybe. But I don't think we're going to see those pressures if we can just supplement with folate, right? Because those people will continue to survive in a sunny environment. I think the other way around, if we have someone with darker skin in a cloudy environment, bottom line is we have less efficient vitamin D metabolism in someone darker skin because they may not have that particular allele of the DHCR7 gene itself. So in that substrate, I think that we can always supplement with fortified foods, and I take a vitamin D supplement just because I think it's good for me. So because we can supplement in different ways, we can wear sunscreen and prevent skin cancer, and we live these long lives, I don't think there's active pressures that are going to change things. I do think that just as over time, as we all mix together and mate with each other, I think we're going to find just mixes of variants, and maybe through just random mating of different genotypes together, we'll solve some of these issues without vitamins. But I don't think it's an environmental pressure at this point that's going to change things. It's more of just a personal pressure. That makes sense.
Yeah.
I mean, you really have to have a pressure on reproduction, which, as you say, it's not going to be the case.
But I would take folic acid if you guys lived in Phoenix and you were fair-skinned.
Yeah, for sure.
I don't know. It's pretty hot there. I think we're safer here, at least for the summer.